All right, let's dive right into something that sounds straight out of a sci-fi movie, but is actually happening right now in the real world. Welcome to this Explainer, where we're unpacking a truly monumental and hotly debated breakthrough in human genome editing. We're looking at a brand new milestone in the quest to edit the genetic code of human embryos. It is complex, it's moving at a blisteringly fast pace, and honestly, it's a technology that could literally dictate the genetic makeup of future generations.
Now, to really grasp why this new announcement is such a massive deal, we gotta rewind a bit. Remember 2018? That was when Chinese scientist He Jiankui absolutely shocked the world by creating the very first gene-edited babies using CRISPR. The scientific community was outraged, called it totally reckless, and he ended up serving a 3-year prison sentence. Okay, now fast forward to June 1st, 2026. A team from Columbia University just posted a preprint paper showing a completely new way to edit human embryos. It's a totally different tech with different goals, but man, it brings all those past controversies rushing right back to the surface.
So here's what we're going to cover in this explainer today. We'll unpack this new embryo breakthrough, compare original CRISPR to base editing, look at the specific target genes, explore a major biological hurdle called mosaicism, and then map out the fierce ethical dilemma of cures versus designer babies.
Let's get right into part 1, the new embryo breakthrough.
So what exactly did they do? Well, a team at Columbia University, led by developmental cell biologist Dieter Egli, has achieved a pretty massive milestone. They didn't just edit human embryos. They are the very first to successfully use a highly precise technique called base editing to pull it off. And this isn't just some minor update to an old method. Experts are calling this a major conceptual shift that could fundamentally push the entire field of genetics forward. Moving on to part 2, CRISPR versus base editing. Okay, this is where the science gets really cool. Think about the first generation CRISPR-Cas9, the tool used in that 2018 scandal. You can kind of think of it like a pair of blunt scissors. It physically cuts both strands of the DNA. And previous studies actually showed that doing this in embryos can cause the catastrophic loss of entire edited chromosomes, which makes it totally unusable and incredibly dangerous. But second-generation base editing, it's completely different. Instead of blunt scissors, think of it as a super precise pencil and eraser. It doesn't sever the DNA at all, it just targets one single letter in the genetic code and rewrites it. It's way more reliable and dramatically lowers the risk of those unwanted genetic mutations. Which brings us to part 3. Editing 3 specific genes.
So what were these researchers actually trying to rewrite with their genetic pencil? They focused on early-stage human embryos and followed a really targeted process. By making just a single letter change, literally swapping the DNA letter A for a G at an exact, specific spot, they were able to fundamentally alter how these genes function. Depending on the gene, this tiny single base swap either completely switches the targeted gene off or it mimics a naturally occurring protective mutation. I mean, we're talking about an incredibly delicate operation here, changing one single letter out of billions.
And this is where you really see the massive medical potential. First up is the PCSK9 gene. By switching this gene off, they mimic a natural state that lowers bad cholesterol. Down the line, that could potentially prevent things like coronary heart disease, heart attacks, and strokes. Then we have HBG1 and HBG2. For these genes, the A to G swap mimics a natural mutation that boosts a protective type of fetal hemoglobin. And the medical goal there is absolutely clear: treating devastating blood disorders like sickle cell disease and thalassemia. You can easily see why the medical world is watching these specific targets so closely. All right, part 4, the mosaicism hurdle. But hold on, before we get too far ahead of ourselves, there's a major biological catch. The edits didn't take uniformly. The researchers hit a massive roadblock known as mosaicism. Basically, this means that when the embryo develops, it becomes this sort of patchwork. Some cells successfully take on the new edited letters, while other cells just keep the original unedited ones. So you don't end up with a fully edited embryo at all, you get a mosaic. And mosaicism wasn't their only headache. The team ran into some pretty severe complications with how the editor was actually delivered. To get the base editor into the cells, they used snippets of mRNA, but at the super high doses required to make the edits, that mRNA literally caused the embryo cells to stop dividing entirely. Plus, the base editors themselves can have other unintended, damaging effects on the embryo. Because of all this, the researchers are heavily emphasizing that this tech is strictly confined to the lab right now. It is absolutely 100% not ready for human pregnancies, though they are already trying to improve their procedures to reduce that mosaic effect. In fact, the lead researcher himself is the one hitting the brakes on the hype. Listen to this. Dieter Egli says, "These base editors, they can have damaging effects on the embryo. So why would you use it if you don't fully understand that? In its current form, you can't use it. It's as clear as day and night." He's making it completely unequivocal. The scientific milestone is very real, but actual clinical application, that is still a long, long way off. And that leads us right into our final section, Part 5, Cures versus Designer Babies. As you might guess, this has sparked a massive ethical debate. On one side, a big chunk of the scientific community is really praising this work. Genomics researcher Greg Neely is quoted saying, "This will go down in history in a positive way, less reckless, more careful, and ethical than previous attempts." For scientists looking to cure devastating inherited genetic mutations like sickle cell disease, they see this as a cautious, well-documented, and frankly necessary step forward to alleviate human suffering. But man, on the exact opposite side of the spectrum, the alarm bells are ringing loud and clear. Biomedical ethicist Hank Greeley points out a pretty terrifying hypothetical. He warns that affluent individuals might just view this study as an open invitation. Somebody with a few $1 million could easily set up a private IVF and genetic testing lab and simply start trying these base edits completely off the grid, far away from strict regulations. And because of those massive hurdles we just talked about, like mosaicism and cell damage, Greeley warns that one result could be, quote, really sick kids. And some experts take the criticism even further, asking if we even need this technology in the first place. Dr. Fyodor Urnov argues that editing human embryos to treat disease is really just a solution in search of a problem. Why? Because couples who carry genetic conditions already have access to standard IVF and genetic screening to make sure they don't pass those conditions on to their kids. Critics like Urnov fear that since we already have ways to prevent genetic diseases, the real, inevitable commercial application of base editing won't be about curing the sick at all. Instead, it'll be fueled by affluent parents looking for baby improvement, trying to edit for superior intelligence or other specific physical traits. And that really leaves us staring down the barrel of the ultimate question. As the science of base editing hurtles forward, moving from those blunt genetic scissors to precise genetic pencils, where exactly is the line between preventing devastating diseases and engineering humans? This technology is no longer just theoretical. It is actively being refined in the lab today. So we have to decide as a society exactly where we draw that line before the technology just goes ahead and draws it for us. Thanks so much for joining me on this explainer. I hope it helped clear up both the incredible science and the incredibly complex debates surrounding our genetic future.